Hello, and welcome to our explanation of metabolism. Metabolism is a set of life-sustaining chemical transformations within the cell of an organism. Via our example, the substrate turns from green to blue to yellow. Catabolic reactions are when larger molecules are broken down either for energy or to be used in an anabolic reaction. From here, the green turns into blue and yellow, and then the blues turn into different shades of blues, and the yellows turn into different shades of yellows. An enzyme is a protein that acts as a catalyst for biochemical reactions. This was difficult to draw, but as you can see, it speeds up the reaction quite quickly. Okay, now let's move on to endothermic and exothermic reactions. Endothermic reactions, as shown by the graph, is when a reaction takes in heat, so the activation energy exceeds the energy formed in the bonds of the product. Without the enzyme, the activation energy needed is much larger, whereas with the enzyme, the activation energy is much smaller. For an exothermic reaction, this releases heat, and so as similar to the endothermic reaction, the energy of activation is much higher without an enzyme, whereas the energy of activation is much lower with an enzyme. This means that an exothermic reaction is when the formation of products releases more energy than is required to break the bonds. An inhibitor changes the shape of an enzyme so that it is no longer in an optimal position to catalyze the reaction. A competitive inhibitor, as shown on the screen, blocks the active site, which prevents any binding between the substrate and the enzyme. Meanwhile, a non-competitive inhibitor binds to another part of the enzyme. The substrate can still bind to the enzyme, but the reaction cannot be completed. This all leads to inhibition. Inhibition reduces the rate of substrate binding or prevents it completely. This ensures homeostasis within the cells by regulating the metabolic pathways. And now presenting our 5-step metabolism pathway. We start with our substrate, Bo, Bob, Joe, Bill, Sam, and our enzyme, which, for further purposes, will be blue and labeled E. Bo, Bob, Joe, Bill, Sam is the initial reactant. Here we can see Bo, Bob, Joe, Bill, Sam connect to the enzyme. Here, Bo is split off from the rest of the substrate. Now we enter step 2, and we still have Bob, Joe, Bill, Sam. Bob, Joe, Bill, Sam is an intermediate. Here we can watch Bob, Joe, Bill, Sam enter another enzyme, and Bob is split off from the substrate. Now we have Joe, Bill, Sam, who is still an intermediate. For step 3, we watch Joe, Bill, Sam enter the enzyme where Joe is split off from the substrate. Bill, Sam is still an intermediate. In step 4, we watch Bill become separated from Sam. Now we know that Sam is the ending product, the final product. Sam continues to be a non-competitive inhibitor of the first enzyme so that this cycle cannot continue. Bo, Bob, Joe, Bill, Sam 2.0 continues on, not split up.